Hello folks, welcome to Tea with Tom again. At this time of year, with Christmas approaching, what could be more exciting than reviewing what's happened with Christmas Factor over the last few years? Of course, Christmas Factor is Factor 9, but perhaps it ought to be Factor 12 for the month of December. Anyway, it's a very interesting story with Factor 9. It was really gained a lot of publicity and interest when in 1952 a young lad called Peter Christmas, who was a Canadian 16-year-old boy, was investigated for haemophilia at the Oxford University Clinic run by Rosemary Biggs and R.G. McFarlane. He of the biological amplifier or waterfall cascade proposal. Of course, haemophilia was well studied at that time, and they carried out a factor 8 assay on Peter, and his level turned out to be 100%. And yet he had all the clinical features of haemophilia, so they thought a new factor. But in fact, oh yes, and their work was published, of course, in the December issue of the British Journal of Haematology. But history has got another record in that Alfredo Pavlovsky in Argentina had already described a different form of haemophilia due to a different clotting factor already back in 1947, when he mixed together his plasmas from haemophilia patients in order to use them for factor assays. And he found when he mixed them all together, they became normal. And they found there were two types of haemophilia, A and B, of course, at that time. So anyway, a lot has happened since that time. With the availability of factor 9 deficient plasma, that allowed assays for factor 9 to be carried out with OSCA one-stage clotting assays. And factor 9 became characterized over time. Its half-life was established longer than that of factor 8. And factor 9 was purified from blood and was available as prothrombin complex concentrate for many years. In more recent years, it was, of course, made by recombinant technology, and, of course, modified forms of factor 9 became available with longer half-life, higher activity. Since that time, we've also had chromogenic substrate assays for factor 9, and, of course, in recent years, genetic analysis of the factor 9 gene has become very well studied. There are many mutations of factor 9, I think 3,000 at last count. Now that we have this technology, people have looked back at Peter Christmas's genetic problem, and it turns out to have been a cysteine to serine mutation at one point in the molecule. Of course, haemophilia has been a fascinating area. The royal haemophilia, which Queen Victoria passed on, and which Tsarevich Alexander of Russia had, turns out to be not a defect in factor 8, but a defect in factor 9, which is a bit rarer and makes it more royal than usual, I suppose. Anyway, the royal mutation, it turns out to be a splicing defect in the factor 9 molecule. Among these mutations, there are some which have a gain of function, which means that the factor 9 is more active than expected from its antigenic level. And these gain of function ones are quite interesting. There's one called factor 9 Padua, which is used now in the gene therapy, which has recently been announced by CSL. CSL had a partnership, or has a partnership, with a Dutch company called Unicure. And over a period of research, they have developed an adenovirus vector containing the gene for factor 9, in this case, the Padua factor 9 mutation, which is about 5 or 6 times more active than normal factor 9. And basically this has been in clinical trials now, and after injection, the patient, 94 of patients in the trial actually showed very good recovery and no further need for factor IX concentrates for a satisfactory lifestyle. So that's been approved now by FDA. Mind you, the treatment costs $3.5 million per shot, which will make CSL have indeed a very Merry Christmas. So, signing off for Christmas. I wish we all at Haemotex wish you a very Merry Christmas, and thank you very much for your interest in blood coagulation and these updates. Thank you. Merry Christmas.